Individuals with long COVID experience more than 200 symptoms, and the different phenotypes or classification of long haulers are not well defined. How are you identifying the most suitable group of patients who are likely to benefit from the use of each of these drugs in your clinical trials?
It's a great question and it's a challenging question. I'll preface by saying all opinions are valid here. I'll tell you what our approach is, and I'm not sure that it's the right one, but it's the one that we've selected. And I think the key phrase here is experimental medicine. We've started our early clinical trials, so we're testing about 7 or 8 drugs or different interventions right now, and we've started our early trials with a broader approach to inclusion, with every single person that is enrolled in the trial being deeply phenotyped. Although we are casting a bit of a broad net in who we include, and to that end, really the only thing that we need for inclusion is a confirmed SARS-CoV-2 diagnosis and a confirmed long COVID diagnosis. But what we do is we deeply phenotype everybody. So we do a full immune panel, including immune testing that can't be purchased or done using traditional bloods. So we work with partners like Akiko Iwasaki. We're doing very detailed neurological assessments. We're doing very detailed epithelial assessments and what have you, so that when we do see responders, we can understand who responds. And also when we see non-responders, we can also see that a biomarker of interest doesn't move. Because I think that understanding why something doesn't work right now at this stage is just as important as understanding why it does. Because if you ask anyone who's either researching in the long COVID community or treating in the long COVID community or just a member of the long COVID community trying to get by, trying things out, they will tell you that some things work on some people like magic, and other people it does absolutely nothing. I think possibly one of the most relevant examples of that would be low-dose naltrexone. You will hear a large swath of people saying, I tried low-dose naltrexone and it did absolutely nothing for me. And you'll hear other people say, it has turned my life around. What we need is deep phenotyping studies to understand, why did the person who didn't respond not respond, and why did the person who did respond? And currently, we're not getting that. Currently, we're getting clinical trials that just give a whole bunch of people the drug. Oh, you got better, and you didn't get better, and we'll just report the statistics. Or even worse, we see case series coming out of clinics where maybe they're treating thousands of people with the drug, but only 100 or so get better, and they publish that case series of the 100 people who got better with no insight into why they got better, no insight into what was the difference between them and the 2,900 people who didn't get better that drove the response. And if we could really dig into that, I think we would start to better understand what's happening in the community.